So today we're going to talk about genomics, transcriptomics, metabolomics, and microbiomics, etc. So I put this title Omics. So we already talked about genomics, and we explained what it is. But it's basically the study of the structure and function of the genomes as a whole. And basically here you are studying DNA, right? DNA sequence. For RNA, we talked about transcriptome, which is exactly the same concept, which is basically all the numbers and the kinds of the transcripts a genome makes. And we generally refer to that study as transcriptomics. So today we're going to talk about a little bit about proteins. So within the same lines, all the different kinds and the amounts of proteins that a species can make, or in a cell or in a tissue, whatever you're studying. So that as a whole is called the proteome, right? And the study of that is called proteomics. Right, that should be pretty clear to everyone. All right, we're going to talk a little bit about proteome. So you have already learned through the semester that there are different ways in which we can study protein structure and function. Some of these basically try to identify and quantify proteins, and some of these techniques, actually all of these techniques, are based on some property of the protein, such as the size, the charge of the protein, or the shape of the protein, right? So one of the first things that when you take a whole different mixture of proteins that you can isolate from a sample is to separate these proteins based on their property. So these kind of separation techniques you've learned were based on centrifugation or based on biochemical columns that can differentiate proteins on their charge and size, etc. So this is an example by which you can actually study proteins. It's a 2D gel, two-dimensional gel experiment. So in this experiment, what you're doing is you have a mixture of proteins in your sample and you want to separate them. So you can study one protein at a time. So what you can do is you can first separate your protein mixture in one dimension based on some property, and in this case the property is based on its charge. So you use a pH gradient to separate all the proteins based on their different charges. And you do this on a gel. So this is kind of like a matrix which allows you the proteins to separate at different locations, right? Very much similar to agarose gels, which separates DNA. So what you do is you then take this proteins separated in one dimension and put it sideways and separate these proteins on a second dimension. What that means is, like, let's imagine this red band here is a mixture of a few proteins that have the same charge, right? Because you only separated them by charge in the first dimension. In the second dimension, what you're going to do is you're going to separate those proteins based on their size. So if this band had two proteins of the same charge, but these proteins were of the same different sizes, they would now travel at a different distance based on their size. So you would find the smaller protein that is running a little bit faster on an SDS electrophoresis page, is an electrophoresis gel, versus a larger protein that runs a little bit slower, right? So you can separate these proteins based on two different properties. So when you do this and you look at all the proteins in your gel that's at the end, this is kind of what you see, which is ugly, right? But what it is every single dot in this gel, 2D gel, is going to be composed of one protein, right? And this protein, if you have a marker molecular weight marker, you can estimate what the size of that protein is and what the charge of that protein is based on the two dimensions that you separated the proteins with. Is this clear? I think you already have seen this before, right? So it should be pretty, it's a kind of a review. All right. How can you use 2D gel to compare two samples? We always talk about this, right? We always want to compare two samples to understand differences between tissues, differences between normal and disease state. So if you wanted to know, for example, what is the protein expression differences between a normal and a cancer tissue, what you could do is you could take these two different tissues, you can isolate them, you can isolate the proteins, then run the proteins on a gel, and then look for different spots, right? Because if you see a different spot in the cancer cell versus normal cell, you know, okay, there is a new protein being expressed in the cancer cell that's not in the normal cell. Or you can do this kind of analysis where you can look at the differential presence of the proteins. What you do here is, for example, you isolate proteins from normal and cancer tissues. And what you can do is you can conjugate these proteins separately to either with the fluorescence dye, CY3, which will label all the proteins green, and CY5, which will label all the proteins red, right? So let's say you took all your normal all the proteins you isolated from your normal sample and fluorescently labeled them with CY3. You took your cancer sample, all the proteins in the cancer sample, and labeled them with the fluorescence of red. And then you mixed them in the same proportion, in the same amount. And then you did the 2D gel analysis. Then what you can do is you can visualize that gel using a scanner that is going to measure the fluorescence amount, red or the green, right? And you can look at this gel, and for each spot refers to each spot is going to correspond to one protein, right? And what you're going to see is that if a spot appears yellow, that means proportionally there were equal amount of that particular protein in the two samples. If the spot appears green, because you fluorescently labeled the normal sample with green, you know that that protein is more expressed in the normal tissue. And if a spot is appearing red, you know that that protein is more expressed in the cancer sample, right? Does that make sense? This is very, very similar to another technique that we actually discussed. What technique is this? Microarrays, right? But in that case, we were analyzing the RNA, but in this case, we're analyzing protein. But the principle by which these are analyzed is very, very similar. Excellent. All right. 2D gel separates the protein so that each spot is pretty much corresponds to a single protein, but 2D gel doesn't tell you what that protein is, right? It just tells you there is a protein running at this spot that's highly expressed in the cancer cell and it's lowly expressed in the normal cell. So next, what you want to know if you are doing this kind of analysis is you want to know the identity of that protein. You want to know what protein is that in that spot. You can also extend this kind of thinking to not just single proteins at single spots, but to understand and to measure and identify all the proteins in a sample. So the proteome of a sample, right? So you might want to analyze the proteome of a heart tissue. And in this case, you can isolate the heart proteins and you can run it on a 2D gel. But all that will tell you is that there are proteins and each spot is a protein, but it doesn't tell you which proteins and how much. So that means you need a method to identify and quantify the proteome. So how do we do this? The technique that the researchers use to identify and quantify proteins is called mass spectrometry. It is basically an analytical tool used for measuring the molecular mass of a peptide fragment. And combined with the genome sequence, it gives you information about the proteome. So now let's go into a little bit more detail. All right. So mass spectrometers, there are different kinds of mass spectrometers, but there are three principles by which they work. They need three components. The first component is an ionization source. That is basically a source from which a charge, which is basically a proton, is transferred to the peptide. So you have a protein mixture. So each protein is going to be charged by this ionization source. So the charged peptide is now called an ion because it's a charged molecule. So these can be a full protein or a smaller peptides, typically smaller peptides. The next component in a mass spectrometer is called the mass analyzer. What it does is it separates these ions, so these peptides, by mass to charge ratios by electromagnetic field. In simple samples, each peptide have a single charge on it. So every peptide in a simple sample is going to have the same charge. So when the mass analyzer separates the samples based on mass to charge ratio, it basically does it based on the mass ratio. Okay? In more complex samples where the charges of the peptides might be different, the story is different. All right. The next component is the ion detector. So this detector basically detects the presence of a peptide. So the mass-separated ions, so these charged peptides, are directed to strike a detector, and the detector measures current over time, basically indicating the amount of signal at each mass-to-charge ratio. All right. So here's a schematic from your textbook, so you should definitely read about this. So what you have here is you have your sample, which is a mixture of peptides, a mixture of proteins, etc., that you place on a metal surface. And then in this case, the ionization occurs on the surface through this laser. So the charged ions, what they do is they accelerate over a magnetic field. And what they do is when they accelerate from the metal target to the detection plate here, right? So this is the detector. So this is called matrix-associated laser desorption ionization time-of-flight Malditov mass spectrometry. Okay, so what this is doing is that it's basically separating the ions based on their mass. And the way it's doing that is it's doing that based on the time of flight. And the principle is very simple. The smaller peptides of lower mass flies faster than the larger peptides of a larger mass, right? That's basically it. So what happens is then the smaller peptides is going to hit the detector earlier than the others. So the detector is going to detect first the smaller ones and then the larger ones. Any questions about this? All right. There are other ways in which you can actually, the mass spectrometers work. This is another one. So this is called the ion trap mass spectrometry. So in this case, the mass of the proteins and peptides are determined by electrospray ionization. So remember, in the first case, the ionization was being done through a laser, and in this case, it's by spraying it out through this electrospray needle. So what you have is you have your samples in a liquid, and you put it in this needle, and this needle sprays it out, and this electrospray ionization basically ionizes each of the peptides, each of the proteins, into in little droplets. So each droplet is going to contain the ion, right? And then these droplets, these ions, are put into the mass spectrometer, where there is a mass analyzer. It traps the ions in an electric field. So then this mass analyzer, what it does is it releases a certain mass of ions at a time to the detector, and the detector detects then how much of an ion with a certain mass is present, right? The more it hits the detector, you know, the more of that ion you have. All right. That's pretty much it. What is the output from mass spectrometers? The mass spectrometer's output is called a mass spectrum, okay? So it looks something like this. It's expressed in a graph that looks like this. So this is called the mass spectrum. So the x-axis of this graph represents the relationship between the mass of the given ion and the number of elementary charges it carries. That's basically the m to z value. And in a simple sample, like I said, where each ion has exactly the same charge, this x-axis is going to indicate the mass of each ion, right? So the x-axis is simply the mass, and the y-axis is the relative abundance of the ions. How many times does the detector saw that ion, right? So in this case, it saw an ion of MZ ratio 568.65 100 times, and it saw this ion less than 10 times, etc., right? So if you have a sample mixture that have different proteins, and then you put this and get its mass spectrum, what you can do is, so basically, let's say you have a mixture of proteins A, B, C, and D, and you have 1 part A, 2 parts B, 3 parts C, and 4 parts D, and you put this through mass spectrum, mass spec, you're going to get a certain mass spectrum. And next time, if you have exactly the same sample, right, with exactly the same ratios, you should get exactly the same mass spectrum. But if your sample varies in certain proteins or ratios, let's say that in your second sample, you have A in 1 part, you do not have B, you have C in 9 parts, and a D in 4 parts, what will happen is that mass spectrum is going to look a little bit different, because you're not going to get the signal that comes from this sample, the protein B. You're going to get a lot more signal from protein C, but you're going to get the same signals from protein A and D, right? So what this allows you to do is it allows you to get a mass spectrum for a certain sample, and you can compare other samples to it. Does that make sense? Any question about this? All right. So this is helpful for some things, but it doesn't help you to identify the proteins. Because in a sample, if you have different proteins, if that sample is very simple and you know what's in it, you can analyze it. But if this sample is complex, such as the heart proteome, and you're going to have many, many of these little peaks, but you don't know what protein this is, right? You know that there is a protein giving that peak, but you don't know what that protein is. So how do you figure that one out? What you do is you do a tandem mass spec. That means you do the mass spec and get the first spectrum, and then you do the mass spec for individual peaks under that. Okay? So what this is in our example again, so you have a sample with proteins A, B, C, and D, and each of these are going to be corresponding to each of these signals, right? So what the mass spec does is it takes the sample corresponding to each of these peaks, right? And then what it does is it breaks it up into smaller pieces, that sample. So what happens is that if you have a protein A, right, that is this mass spec has detected, it takes that and it randomly breaks it into smaller fragments. Okay, so because you have multiple protein A's, hopefully in your sample, you're gonna have different peptide fragments coming from that protein. Now, what it does is it takes all of these peptides, right, which is basically, again, a collection of different ions, and it's going to repeat the mass spec again. And it's going to generate a second mass spectrum. And in this second mass spectrum, you're going to have the signal coming from each of these broken peptides. Does that make sense? Now, depending on what the amino acid sequence of that protein is, it can only generate a certain population of different peptides, right? So if your protein is a 100 amino acid protein and you know the sequence of that protein, then you can only break it a certain population of peptides with different sequences. Now, what this then what that means is if you know the sequence of your protein, right, you will know how many different types of peptides that protein can generate. And for small peptides, you can actually know exactly what M to Z ratio that peptide is going to give, right? And then what you can do is you can match this mass spectra to a certain peptide sequence. So in this case, this mass spectra is matching to this sequence, right? So if you have a database containing all possible mass spectrums coming from all different kinds of peptides, what can you do? What you can do is, once you run your sample, you get a whole bunch of different mass spectrums, and you go to the database and you say, what amino acid sequence does this mass spectrum come from? And the database is going to tell you, okay, that particular mass spectrum can be generated from this sequence. Yeah?
How do you know that's the particular sequence, though? Like, if you swap some amino acids around?
It will change. The mass spectrum will change. Yep. Each peptide is going to give you a particular mass spectrum that's going to be different from other peptides. In fact, you can even study post-translational modification of proteins, because each peptide, if it's phosphorylated or acetylated or methylated, etc., the mass spectrum is going to change in a certain way in which you can detect that kind of difference, right? So each peptide is going to have a unique mass spectrum. And then what you can do is, once you know the sequences of these amino acids, you can go back to the genome and you can say which protein is encoding for this amino acid sequence. And then you can say, okay, that protein is TBP or whatever, etc. Now what you did is you matched the mass spectrum from your sample to a certain protein, right? Does that make sense? Okay. So that tandem mass spectrometry is typically what you would use to identify the proteins in your sample. So this mass fingerprint, basically here, this is mass spectrum. It's also called a mass fingerprint for a peptide, matches to the peptide of sequence this, and that's very key. So just to summarize what's going on here, right? You basically start with a complex mixture of proteins. You break the proteins into peptides, and then you further break the peptides to get a mass fingerprint for each peptide, right? So this part is what is tandem mass spectrometry is doing, right? Then you match the mass fingerprints that you obtained to a database to determine which proteins can produce the set of mass fingerprints that you observed. So then that lets you identify the proteins in your sample and how much, right? If you see a certain population of peptides many times matching to a certain protein, you know that that protein in your sample was abundant, right? But if you see one or two peptides that match a certain protein, but only a few times, that you know that protein in your sample was less abundant. Does that make sense? All right. So basically what that means is the number of times you detect a certain protein in this analysis would be proportional to the number of measurements you make, right? Number of measurements, and that would be proportional to the amount of protein in your sample. All right. So this type of workflow should remind you something immediately. So what transcriptomics techniques does this workflow resemble? Speak up. I hear the right answer. Somebody is guessing it. Speak up. Come on, guys. What does this resemble in transcriptomics? What is it? Mm-hmm. That's not transcriptomics. You're not analyzing RNA. Yes, thank you. Who said that? Thank you very much. That's exactly what it is. So you basically start with a complex mixture of RNA, right? So that's your resemblance here. And then what you do is you generate your RNA-seq library, then you sequence the library, and then that generates short reads. What does those reads resemble to you? Those reads should resemble to you mass fingerprints of each peptide. Then you take that sequence and map back to the genome, which is exactly what we're doing. You take the sequence of the peptide, map it back to the genome, and find out what protein does it come from. And in RNA-seq, the number of reads that corresponds to a gene is proportional to the amount of RNA that gene was transcribed into in your sample, right? And in this case, the number of peptides that you see from a protein is proportional to the abundance of the protein in your sample. Is that clear to everyone now? Any questions? All right. So that was basically a very simple, straightforward proteomic analysis. With this, you can basically identify and quantify the different proteins in your sample. But to analyze the data, you actually need the genome sequence, right? So you need to know in your genome what kind of different proteins does that genome can actually encode for. So there are other ways to there are other omics techniques in very similar terms, analyzes the numbers and the kinds of things in your samples. One is called the metabolome, and the other one is microbiome. So this basically analyzes all the metabolites in your sample, and this analyzes all the microbes in your sample. And what kind of sample am I talking about? We're talking about mostly humans or some other organisms, right? But I'm going to use the humans as example. So basically, the humans generate thousands of different metabolites coming from different metabolic pathways that are happening in our body, right? And these metabolites are basically telling you the amount of these metabolites and the kind of metabolites are telling you how well these pathways are running in the body, right? And there's some metabolic diseases. For example, one of them is the famous diabetes, right? And it was known for many hundreds of years ago that the patients that have diabetes basically had a sweet taste to their urine, right? And you could basically diagnose the illness based by measuring the metabolite in a sample, which is basically the urine. So you might want to ask yourself, this taste sounds pretty telling that somebody actually is tasting this, right? But that's basically exactly what it was. So in 1674, an English physician was the first in modern medical literature to observe this relationship that the diabetes is basically screwing up the metabolites, that you can actually diagnose that. So here's a quote from the publication. He says he may have enjoyed the sampling process a little too much, stating that the pea on his palate was wonderfully sweet as if it were imbued with honey or sugar. So I'll leave you with that. But of course, the doctors and us, we don't want to taste urine anymore, right? But we need other techniques to actually measure metabolites. And so what you want to do is you want to basically analyze the metabolome, metabolic profile of sample with other methods. And these methods are such as nuclear magnetic resonance, NMR spectroscopy, or mass spectrometry. All right. So people do this. So biochemists and chemists analyze all the metabolites in a sample. And for all the different metabolites that a human can make, these are actually, again, once again, the importance of databases basically are stored into the human metabolome database. What it is that it basically contains and links different types of data that is related to metabolomes, right? So it could have chemical data, which actually gives you the structure of each metabolite. It can give you clinical data, that is what type of metabolites are linked to what kind of diseases. And it can also give you molecular biology and biochemistry data, that is what are the enzymes that are involved in producing or using this certain metabolite, right? So right now the database contains about 42,000 different metabolites, and that's amazing, right? So basically now we have a catalog of all these metabolites, but what we don't know is just like human genome sequencing. When you sequence, you have the sequence information, but you don't know what it means. It's very similar to this case where we have know all the metabolites almost, but we don't know how these are regulated, right? Just to give you an idea what these kind of metabolites are arising from. So they are arising from different pathways in our body, right? So this is a screenshot from the human metabolome database, image showing the glycolysis, glucogenesis pathway that's found in humans. And each of these little squares here that is contain a chemical structure refers to one metabolite that is known to be produced or used within this pathway. So each of these metabolites are basically placed in between the proteins, and they're also placed at different sites in the cell, because the researchers know that these metabolites are used here in the mitochondria, etc., right? So there's a whole different set of metabolites that are being produced through different pathways in our bodies, and there are a bunch of metabolic diseases, so it's really important to know how these metabolites are regulated. Similar story with microbes. So this is just to give you an idea what I mean by microbes in humans. So this is a field that has been kind of exploded in the past 5 years or so. It's called microbiomics. This started all with the observation that the humans, us, are basically a great host for a great number and variety of microbes, right? In fact, if you take and try to estimate the number of human cells in an average human, which will be 10 to the 13, the number of microbes in the same human would be about 10 to the 14. So we contain many, many more microbial cells than we actually contain our own cells. Of course, our own cells are actually bigger so that we don't look like a microbe, but we do have a lot. And if you actually try convert that into some kind of estimate of how many pounds of microbes each of us actually carry, for a 150-pound human, that would correspond to somewhere between 1.5 and 4.5 pounds of microbes, right? Pretty cool. So what the researchers said that, okay, that's an excellent observation. Let's now look in a given human, let's again catalog the different types of microbes that we actually see. How can we do this, right? So what they did is they said, okay, we have all these DNA sequencing techniques. We can actually sequence the DNA of the individual microbes and determine what microbes are there, right? So what they did is they recruited 242 healthy U.S. volunteers, this many males and that many females, and then they collected tissues from 15 body sites, right? So these are not very invasive collections, so it basically was tongue and your nose and et cetera, right? And now what they did is they isolated DNA from each of these samples. So they had about 5,000 samples, and they sequenced the DNA. But what they did is instead of sequencing all of the DNA, which nowadays some researchers do for other reasons, but for identification reasons, what you can do is you can choose to only sequence 1 or 2 genes that would be most diagnostic of what species is there, and in this case that would be the sequence of the 16S rRNA gene, right? So that gene is actually tells you what kind of microbes or what group of microbes are in the sample. And so after that study, they found that there were at least 10,000 different species living in a human body, right? So that was published a few years ago. Basically, this was a work that was funded mostly by National Institute of Health, called the Human Microbiome Project Consortium, just like the Genome Sequencing Consortium. So it basically is a study of the structure and function and the diversity of the healthy microbiome. So this was all healthy individuals that were studied. So what did the researchers find? So in this example, what I'm showing you is a clustering analysis. We learned about how clustering works in microarray examples. So what this data is plotting here is that each of these little dots is the sequencing data from a given sample. Okay? So each sample is going to have multiple 16S sequences, right? From a given sample means from a given one person. And then what they said is, for that set of 16S sequences that I see, look at all the other samples and plot whichever is the most closely resembles, so whichever looks very much similar, and plot that right next to it, okay? And then it did that in many iterations and separate the data into the 2 dimensions. What this means is that all the samples that are shown here had the similar collections of 16S sequences that are coming from, and those samples were different from the 16S sequences in these samples here, right? And then what they did is they color-coded all of the samples. So they color-coded so that they separated the samples from where in the body that they were collected from. So these samples were collected from gastrointestinal samples, the greens were skin samples, the red was nasal samples, and the orange was oral samples, etc. And what you can see immediately from this data is that at our different body parts, we contain a different sets of microbes, right? So the sets of microbes in the oral samples were different from the sets of microbes in the gastrointestinal samples. Does this make sense? Clustering makes sense? Okay. So you can analyze the data in a different way. So this was basically asking for each given sample from a person, let's analyze all the different microbes and group them based on where we picked these samples from, right? In this case, what they're saying is that now, let's do the analysis in a different way. What they're doing is that they're looking at 127 tongue samples, right? And then they're saying, in those samples, what we see these type of species, right? And they say, okay, we're going to color code each species. So you have Streptococcus sanguinis, or however you pronounce it, etc. Right? So in the tongue samples, they are taking all the Streptococcus sequences, and then they are color coding it based on what species it's coming from. And then here, what they're doing is on the x-axis, they're plotting each sample from 127 tongue samples. So these are basically coming from 127 different individuals, right? And in the y-axis, what they do is they plot the relative abundance of each of these species. What that means is that, look at this one, Esperan sanguinis, the blue, in this sample, a very large proportion of the sample from that person contained this particular microbe, and it contained a little less of everything else. In this person, the tongue sample contained not so much of this particular microbe, but it contained more of the other microbes, right? And this doesn't add up to 100% because in the samples, it's not just the strep bacteria, there are other bacteria present as well. But what should strike you, which pretty much strike everyone, is that given an individual healthy person, there is a lot of variation between the amount and the types of bacteria that person can have on their tongue, right? So that the variation between person is pretty great. All right, and that variation we need to understand because when you do these kind of microbiome studies, now comparing healthy people to for example, people who have a strep throat, you want to know which of these measurements are due to variation and which of these measurements might be actually due to that person being sick, right? All right, so this kind of microbiome experiments are awesome. They're really fun, and you can do it anywhere. And in this example, what Chris Mason and his colleagues have done is they said, okay, let's look at the microbiome in the New York subway stations. So he had a few students, and these students, what they did is they took little swabs, and they swabbed a lot of turnstiles, ticket kiosks, railings, and benches, etc., right? And now what they did is they isolated the DNA and sequenced the 16S and basically mapped the different types of bacteria and viruses, etc., that are known. And they also found that there are lots of sequences that they could not detect in the databases, right? If the database doesn't have the information for a given species, you're never going to find that species, right? You might get the sequence from that species, but you won't know what that species is. And if you map that into now the subway map, you can see the different kinds of bacteria. Where we live seems like it likes lots of bacteria, right? So that's basically the type of analysis you can do. What do you want to use this for, right? Is this a waste of time? I mean, some researchers might say yes, but others might say no. But if you actually could do this kind of analysis sporadically, and on the subway, what you might be able to do is you might be able to, for example, catch epidemics or things like that earlier than maybe with other methods, right? Because you'll catch the species immediately on the subway because everybody uses the subway, right? So not only it's fun, that it can have future applications to how we live. All right. So far we basically saw the different omics techniques. That means genomics, transcriptomics, proteomics, metabolomics, microbiomics, right? And these are done can be done with many different samples, which are basically people, right? So that brings me to what I would call personal omics. And why is this important? It's mostly important to understand the humans as a population, and also it has some applications for medical fields, right? So what this type of work does is that if you know the personal transcriptomics or etc. For a given person, what it can allow you to do is if you have analyzed many people, it can basically allow you to compare the genome, transcriptome, proteome, microbiome, metabolome, etc. of two groups, for example, with or without disease, and find out what distinguishes these two, right? And so it will serve as a starting point to test if the differences that you found are causal. So if a difference that you found is causing certain thing that you're analyzing. For example, in theory, we should be able to sequence everyone in the world, right? and find the DNA sequences then underlying the differences in our phenotypes, right? However, it's not that simple. It turns out it's not that simple because what we don't know is that this requires knowing the mechanism by which the genome works. So we have to supplement this with a lot of the functional studies of the genome to understand what these differences mean, right? So what do I mean by differences in the sequences? So how actually similar are we? It turns out we're very similar to each other, but we're also very different, okay? So we're very similar because on average, two random people share the same DNA sequence in 99.9% of their genome, right? But we're also very different because we have 3 billion base pairs in the genome. So that also means with the same calculation, on average, two random people will differ at 3 million base pairs. So that's 1 in every 1,000 nucleotides or so. So what kind of differences are we talking about in the genome? So these differences can be classified into several groups. One is called single nucleotide polymorphisms, called SNPs, right? Those SNPs refer to the different single nucleotide differences at a given location in our genome, from one person to the other person, etc. So these could be just the changes, or it could be a simple deletion of one nucleotide, which is called an indel. The second type of difference that we actually learned in our DNA lecture, one of the very first lectures, is the simple sequence repeats, right? So these are microsatellites that are found in different copies in individual people. And another type of variation that the humans have is copy number variation. That means there are regions of the DNA that are 1 kb to several megabases that are present in different copies in each of us, right? So one of us might have the same gene in 2 copies. One of us might have the same gene in 4 copies if it's duplicated somewhere else. All right. So since we know that the genotype is affecting phenotype, if we knew the genotype and if we knew how each of these sequences actually function, then we could understand the phenotype. All right, so how these polymorphisms actually appear? Polymorphisms means the differences, right? These polymorphisms appear due to mostly errors in DNA replication and errors in repairing of the DNA. So polymorphisms result mostly from replication error and unrepaired DNA damage, and they're inherited if it's present in the germline, right? So most of the SNPs actually occur in introns and non-coding DNA. They do affect function, but probably to a lesser extent of the SNPs that occur in the coding regions of the genome, because it can immediately change the amino acid sequence of a protein, and if the amino acid sequence of a protein changes, it might change the function of that protein, right? All right. So this brings me to how polymorphisms segregate in a population, right? So we basically, for a given region of the genome, so it's shown here just along the x-axis, it can contain several polymorphisms. So these polymorphisms here have been donated with one of these colorful tick marks and with a identity, right? So it's this SNP A, SNP B, SNP C, SNP D, etc., right? And so they can be in the non-coding regions, and they can be also inside the genes. So in the human genome, these groups of SNPs are separated by recombination hotspots, okay? So these hotspots are regions on the chromosomes that most crossovers occur. What does that mean? What that means is that there is not going to be very much recombination in between two hotspots, okay? So because there's not much recombination between these two hotspots, these group of SNPs here, A, B, C, D, E, are going to be passed from the parent to a children as a unit, right? So this is called a linkage disequilibrium block. So this these group of SNPs are going to be segregating in the population together. If there was crossover equally across every site in the genome, then these SNPs wouldn't have segregated together. So a haplotype is basically the sequences at these SNPs, A, B, C, D, and E. All right. So, furthermore, if a certain gene X basically causes a particular trait or disease, a certain haplotype, so basically this particular sequence at these SNP sites, will correlate with that disease, right? So this is the basis in which the genome-wide association studies are performed. So here's an example. There's actually a very nice study question about this. So if this doesn't make sense to you, make sure you go back to the textbook, read the chapter, the relevant chapter, and go to this study question and see if you can figure this out. Because if you can figure this out, that means you pretty much got the idea. All right, here's an example. So here's a genome-wide association study, theoretical experiment. So I divided you guys into two groups. So into all the students who told me that they love MCB, and all the students they told me that they live MCB, right? And then I sequenced in each of these students. So at a given site in the genome, what you can find is for all the individuals here, 1, 2, 3, etc. So you're going to have a SNP location. In this case, it's SNP1, SNP2, SNP3. And that SNP location, the sequence of between individuals are going to differ, right? So this person is going to have a C in this location. The other person is going to have a G, etc., etc. So at the SNP2 and SNP3, etc. So then I look at group 2 at these certain loci, SNP locations. And what I see is that, okay, for this location, I see a similar distribution, maybe. And in this location, interesting. There's not that many C's in the students who live MCB. And in this location, there's not many A's in students who live MCB, but the students who love MCB have a lot of A's, right? So what's the haplotype then associated with loving MCB? The haplotype is the set of sequences for a given linkage disequilibrium block, which is SNP1, SNP2, and SNP3. What is the sequence that is most associated with loving MCB? Just tell me the sequence of this location, and this location. Is it CGA? Is it CCA? Is it GCA? Probably, right? So because what you can see is that the A is very common for students who love MCB, but it's not so common here. C is very common for students who love MCB, but it's not that common here. So what you can tell me is that, this haplotype is associated with loving MCB. What this little thought experiment is not telling you what is actually variation and what is correlation, right? Because in this toy example, I only showed you a few individuals, right? So already you can see at this spot that all of the group 1 contains the C, but there's a person in group 2 also contains the C, but that person is not loving MCB, that person is living MCB, right? So what that means is that the causal, which of these SNPs are actually causing the phenotype, to be able to tell that, you need to know the variation of that SNP at a given locus, right? What that also means, to know what the variation is, you need to sequence many people to know what the population variation is, right? So then GWAS, which is Genome-Wide Association Studies, that need a very large number of individuals to be able to tell which SNPs are actually associated with a disease or a phenotype, right? In this case, in the study, what they looked at is they took about 9,000 individuals. They genotyped it for a certain number of SNPs. So instead of sequencing the whole genome, they said, let's just look at the sequences at the SNP sites that we know are different in between different people. And then they looked for 22 common traits like hair curls, freckling, eye color, etc. And then what they do is they find SNPs and genes associated with 22 common traits. And the way that they plot this data is called a Manhattan plot, because it's kind of like these blocks of buildings. So what the Manhattan plot plots is that it tries to highlight the genomic region that associates with a given trait. All right, so how's that work? Basically, on the x-axis, what you have is each chromosome, right? So chromosome 1, 2, etc. And each of these dots is a SNP location that was tested, all right? And on the y-axis, what you have is you have a p-value, which is basically an estimation of how well the certain SNP is correlating with the disease, okay? And what it does is it basically, it's plotted on a negative scale. So if the p-value here is higher, that's going to tell you that SNP might be associating with that trait that you're looking at. So here, those SNPs that were basically found to be significantly associating with the trait is found on chromosome 2 at a certain location, right? And so that was for curly hair, and this was for freckles, right? So what they found is that there are multiple sites in the genome, right, on chromosome 6, chromosome 9, etc., where a number of SNPs were associated with that phenotype. This doesn't mean that these SNPs are causing the phenotype. What that means is that these SNPs are at a region of the genome whose sequence might be causing the phenotype. Okay? So that's a distinction that you should really, really try to understand. So this type of genome-wide association studies basically has been exploded in the past few years. The number of studies are constantly increasing because people want to know what the genotypic basis is for certain phenotypes. But although it's been very fruitful in some cases, in few cases, it has been also not so fruitful for a lot of the cases. Because after so many studies, what the field has recognized is that there's actually a one gene, one trait type of situation is found for only very few genes. So for one gene type of diseases, these type of studies have been very phenomenal. But for a lot of diseases or traits, such as human height, weight, heart attack, obesity, etc., these traits involve many, many more genes. So then when it involves more genes, what happens is that the certain SNPs that are associated with a trait explain very little of the variability of that trait. What does that mean in plain words is that you can carry a certain SNP, right, that is known to be associated with a certain trait. But you having that SNP is not predictive. What that means is that you having that SNP, you will be almost equally likely or not to have that disease. So that SNP information, even though it is associated with the disease, is not predictive if you're going to have that disease, okay? Because many, many different SNPs are associated. Sometimes you having a certain SNP at this site is canceled by you having another SNP at another site because these two genes interact, right? And you having a certain mutation is basically overcome with a secondary mutation that actually helps alleviate the problem. So that is basically the problem with a lot of these Jiva studies. So what that means in this example I just gave you, right? That mutation in one side is being compensated by another mutation on another side. You can actually infer these if you knew that the sequence at one side of the genome is functionally linked to another sequence at another side of the genome. What that means is that you need to know how the genome functions. You need to know what part of the genome is interacting with another part of the genome. And when I say what part of the genome, I mean it can be genes, it can be regulatory sequences, right? It could be a lot of different ways in which different sequences in the genome can interact. All right. So back to our toy example, in which we said, okay, this haplotype here is associating with the loving MCB, right? And if I ask you then, which SNP causes loving MCB, your answer will be, I don't know, right? Because you don't know if the SNPs that you see here, for example, A, is causal, that means it directly causes the phenotype, or that it is basically linked to another sequence that is close by within that same linkage disequilibrium, right? Because they're segregating together. So what I mean by that is, okay, you see that, all right, A is associated with loving MCB, right? But there is some other sequence here that, for example, that you don't know because you never sequenced or you actually don't know. That might be within a gene X that is surrounded by by the hotspot, crossover hotspots. So this whole thing is basically traveling together. It's being inherited together, right? Even though you know that this A is being associated, the causal SNP might be here somewhere that you didn't actually look. Does that make sense? All right. So when you did this experiment and you found a SNP that really associated, I mean, this would be absolutely striking for a Jiva study. This almost never ever, I would say never ever happened, and probably never happens, right? So once you found this, you need to then distinguish what is linked and what is causal. How would you do that? So if you were a researcher and you were very fortunate that you found a certain SNP that very highly correlated with obesity. Everyone wants to lose weight. You can make billions of dollars. You really want to follow up this SNP and find if that's causing obesity. What would you do? And you know that genomic region, right? I just told you. There is a region of the genome, let's say that's 10 kb, and it contains that certain gene named gene X. And you say, because this SNP is very close to gene X, and it's basically traveling with all the sequences that are different between individuals that are on gene X, I'm going to hypothesize that the gene X is linked to obesity, and I'm going to now study this, and I'm going to study the different sequences that I see in gene X between skinny people and fat people, etc., right? Where do you start? How do you attack this kind of research question? Come on, fire your imagination. We have 10 minutes. Billions of dollars at the end of it.
You studied in model organisms?
Exactly. We talked about models constantly, right? You could try, because you can't really do a lot of experiments in humans, Jiva studies basically serve as a starting point, right? So what you can do is you can say, which model organism actually has that gene X, right? Let's say the rats had it. You go to the rat model and you say, okay, I'm going to take the gene X in rats and I'm going to look at the variation of sequence and how gene X sequence and how that affects the rat weight, right? What can you do to do this? So let's say on gene X in humans, you found a certain mutation in an exon of a gene X, right? That was associated with obesity. And you want to test if that certain sequence is actually causing obesity. Being associated doesn't mean it causes, right? Associated means correlation. It's not causation. How do you actually test if it causes obesity in your model system?
Do a knockout.
Well, if you knock out, what you will know is you'll know if that gene is important for any type of obesity-related phenotype, right? It could affect sugar metabolism, it could affect weight, etc., etc. And that's something the researcher will probably do first. But I'm asking the question, for a certain given change that you observe in human population, you want to know if that change is causing obesity.
Use something like CRISPR that is.
Exactly. We talked about CRISPR, right? And we said how you can use CRISPR to target a certain site in the genome and change the sequence at that site by providing a homologous recombination template that contains the mutation or whatever change you want to make. So what you can do is you can create a rat that contains exact same variation that you see in the humans, right? So that certain mutation that you see in humans that's associated with obesity, and see if you make a rat that contains that single change as opposed to its clonal sibling that doesn't contain that single change. So you have two genetic backgrounds that are identical, but you're making one change. Then you say, do I actually see that rat becomes now obese? And if it does, then you have a very, very strong proof that that single change is actually causing the disease, right? Does that make sense? All right, that's pretty much along the lines in which a lot of medical researchers are actually working with these GWAS data. All right, so what do we actually do with the GWAS data, right? Of course, whenever there is some basic research and there is some applied potential for this, the researchers actually start companies, and sometimes not the researchers, but other people who know nothing about research also start companies, and they try to use this information, right? And for example, in this case, this is a company called 23andMe. It formed several years ago, recognizing the fact that the sequencing is amazing, and you can actually learn a lot about people and phenotypes by sequencing different people. So what they said is, now we're going to make a genetic test in which we're going to sequence, you're going to send in your sample, and they're going to sequence certain genes that are that contain SNPs with associated traits, right? So an example would be, a very good example would be BRCA1 and BRCA2, right? If you have breast cancer in your family, maybe you might want to get these two genes sequenced because there are known mutations that are known to increase your risk for cancer, right? But GWAS studies actually weren't that far, and they're still not that far. Most of the GWAS studies, it tells you there's a SNP associated with a disease or a trait, but they don't know actually if that SNP causes the trait or how closely it's linked with that trait, right? So this company, basically what it does, it created a genetic test before that information was available, right? So, but then they were giving false medical predictions, right? Because it was saying, okay, you have the SNP, you have a chance of being obese, or you have a chance of a heart attack, or et cetera, et cetera. But like I said, a lot of these SNPs coming out of GWAS studies are not very predictive, right? So the FDA then said, luckily, wait a minute, right? You are actually telling people information that's not really backed up by science very much, right? And so if the patient is not very informed about Jiva studies, they might freak out and say, I'm going to have a heart attack, put me on Lipitor or whatever, right? So, and that would be unnecessary intervention. And they said, okay, you got to stop selling this. And so they did stop selling it in 2013. But the FDA actually allowed them to sell a personal genome test without health-related results, right? So because the 23andMe does this very interesting thing, it looks for your ancestry and then looks for like where part of your genome comes from, what percentage you are Neanderthal, etc., all that fun stuff. They said, do the fun stuff, it's cool. But then in 2015, the FDA said, okay, now we have some information for about 30 or 40 SNPs that are reliable. Then you can take those SNPs and now provide health information for that. And so those reliable SNPs, some examples are basically cystic fibrosis, sickle cell anemia, and Tay-Sachs, etc., right? So these diseases that we know are very well studied. All right, that's it, and I see you tomorrow.